So now is a different discussion that's called membrane potential. All plasma membranes have electrical activities, the movement of ions, but the ones that are important are neurons primarily, but also the muscles. Muscles also have electrical activities, so they respond to the neurons. There is three different type of membrane potential. One is the resting membrane potential. It means that the neurons are not stimulated, so nothing is going on. There is no stimulus there. There is no electrical changes happening. The second one is called graded potential, and in graded potential there is a stimulus arrives in the area, so the membrane potential changes. Okay, so the graded potential happens in the dendrites, in the cell body, in the axon hillock, and also here in the postsynaptic membrane happens here. The third one is called action potential, and action potential happens in the axon, so it transmits a long distance, and also it can happen in the plasma membrane of the another neuron, but especially in the muscles here, so in the postsynaptic membrane of a muscle. If it is a neuron here, it will not directly happen in the connection, in the synaptic connection, because the end of this axon will connecting with the dendrites here. So dendrites will have a graded potential, and then again cell body, and then it can have another action potential. So if it is just a neuron, it will have a graded potential here, and if it is a muscle here, then it can have an action potential. So it depends on the type of connection. It will have a different type of membrane changes. This is something that you may know from Bio 1, but it's good to review it. So this is a plasma membrane, and if you remember it, plasma membrane is a semi-permeable membrane. The outside of that is called extracellular fluid, and inside is the cytosol, so this is the fluid inside the cell. The plasma membrane may have different channels here, and these channels are made of proteins, and you also have some other proteins attached to the plasma membrane. So what happens here, that outside you have a lot of sodiums, a lot of sodium ions, so they are positively charged, and a lot of chloride, which is negatively charged. Inside the cell, you have a lot of potassium ions, which is positively charged, and a lot of proteins that are negatively charged. Okay, so you have these channels, these are called leak channels, that allow movement of these ions. So normally the potassium is leaving the cell, and here is the sodium channel, the leaky channel for sodium, that allowing sodium to enter the cell. Why? Because inside the cell there is not a lot of sodium, and outside of the cell there is not a lot of potassium. But you have something else here that's called sodium potassium pump, or ATPase, that hydrolyzing an ATP to ADP and phosphate, so it uses energy, and what will do is that it brings potassium back to the cell, so 2 potassium back to the cell, and kicks out 3 sodiums out of the cell. So because of this thing that happens, you have a membrane potential, which is normally at rest when there is no stimulation is around -70. Remember, when there is no stimulation, and the ions, the charges inside the cell and outside of the cells are a little bit different. Inside the cell, you can see this is negative here inside the cell, because potassium can leave, but these proteins that are really big, they cannot leave. So the proteins are negatively charged and remain inside the cell, and that's why inside the cell is negatively charged. And outside of the cell, there is a lot of sodium there, and some of the sodium can enter, but they are kicked out again by sodium potassium pump, and this is why outside of the cell is positively charged. Okay. So if you look at it here, you see that the sodium channel, and then in the next slide you will look at the potassium channels here, okay, and the potassium and sodium ions. So here the sodium ion, there's two things to understand. One is the chemical gradient, and the other one is the electrical gradient. So for the sodium, let's talk about the chemical gradient. So there is a lot of sodium outside of the cell, not enough sodium inside the cell. So what does it mean? It means that a lot of sodium try to enter the cell. It's a chemical gradient. What about electrical gradient? Sodium is charged positively inside the cell because of the proteins charged negatively. So it's a high electrical gradient for the sodium to enter the cell. So the net sodium electrochemical gradient is very, very high. So sodium really tries hard to enter the cell. There's something else is here, okay? That is, if these channels are not there and you allow these ions to enter or leave the cell, okay? So a lot of sodium that were outside of the cell now try to enter the cell. They try to enter the cell, okay? And once they try to enter the cell, the inside the cell become more positive until a time that there is no movement of the ions outside of the cell or entering the cell. So there is a balancing of the movement, so there is no net movement. And that is called equilibrium potential. And for sodium, it is positive 66. Remember, the resting membrane potential, when you have these channels and you have sodium potassium pump, is negative 70 millivolt. But the equilibrium potential, if you leave the ions to move freely and then it balances out between inside and outside of the cell, it becomes positive 66. Now let's look at what's happening with the potassium here. So for the chemical gradient of potassium, inside the cell, a lot of potassium. Outside of the cell, not enough potassium. So potassium tries to leave the cell. What about the electrical gradient? So the electrical gradient outside of the cell, it is positively charged, and the potassium is also positively charged. So there is not a lot of electrical gradient for the potassium. So this is why the net potassium electrochemical gradient is small. It's not that high. Although inside the cell is negatively charged, okay, so the potassium is positively charged, so the potassium enters the cell. If you're thinking only about the potassium, but there's also sodium normally there, so this is why the net potassium electrochemical gradient is not as much, okay? But the potassium has about 25% more possibility to leave the cell compared with sodium. Why? Because the potassium channel is leakier than the sodium channel. So it allows potassium 25 times more than the sodium to leave the cell. And if you are thinking of the equilibrium potential for the potassium now, if you let the potassium to leave or enter the cell until it balances out, it will be negative 90 millivolt. So a lot of potassium will leave, okay? So inside the cell will be more proteins, because proteins cannot leave, so the proteins will be there, and that will create more negativity. So it will be negative. And if you compare that with the resting membrane potential, which is negative 70, so the equilibrium potential of the potassium is much closer to resting membrane potential than the equilibrium potential of sodium, which is positive 66. So why the resting membrane potential is not negative 90? Because some sodium still enter the cell, and that will reduce the negativity. It makes it more positive, a little bit more positive. So this is why you have -70 because of the effect of sodium. Here we are talking about one ion at a time, but in real life this is not just one ion. You have sodium and potassium together, and you have sodium potassium pump, and that's why you have the -70 millivolt. So there is a change in the transmembrane potential can happen, and that change can be because of the opening and closing of those channels. And once those channels are changing, the the opening and closing of those channels will change. That is the effect of the permeability of the membrane. The membrane, the plasma membrane, is made of the lipids. So and this is why the ions cannot pass through that. So the ions need channels to pass through that. So once those channels open and close, then the permeability of the membrane changes, and the transmembrane potential also changes, becoming more positive, more negative. So for that, you need to think of the sodium and the potassium channels, because there's other ones also there, but these are the big ones. So these channels are divided into passive and active. The passive ones are also called leak channels. They're always open, and the potassium tries to leave the cell, and the sodium tries to enter the cell. And as I said before, potassium leak channels are 25 times more possibility to allow potassium to leave the cell compared with the sodium channels. And the active channels are normally closed. They are called gated channels. If there is a stimulus, they open, the resting membrane potential changes, and then they close again. So they need ATP. The passive channels do not need ATP. Okay, so there is 3 types of gated or active channels are there. One is closed, but they are able to open. So it's like this, okay? They are closed normally. If there is no stimulus, does not allow any ions to enter the cell. And once you stimulate it, then they can open up. Then there is an open one that is allowing the ions to enter the cell. And the third one is the closed but cannot open. So here is this one. So once the ion enter, after a while it will close, and this is a different gate, and it cannot allow more ions to enter the cell. So there's 3 different states for gated channels. The classes of the gated channels, okay, one is called chemically gated channels. So this is the plasma membrane, and this is the gated channel, which is a transmembrane protein. It has a receptor, and that receptor is responsive to a neurotransmitter. So it's acetylcholine here. Just an example is response to that. So if the acetylcholine comes here, which is a neurotransmitter, the gate opens up and the ions enter the cell. And once there is no stimulus there, then the gate closes back. So it prevents movement of ion to the cell. The second one is called voltage gated channel. Okay, so here is at rest, there is no stimulus there. You have 2 types of the gates. One is activation gate, which is normally closed at rest, and the other one is inactivation gate, which is normally open at rest. So when there is a stimulus comes there, okay, so see that there is from -70 now is becoming -60. So it will open up. Then the ions enter the cell. Once the inside the cell become really positive, see this is positive 30 millivolt, then the inactivation gate will close. This is inactivation gate and prevents further movement of ion to the cell. The third one is called mechanically gated ion channel. So this is a protein. This is a plasma membrane. If there is no stimulus, nothing happens. If you press it, okay, physically press it, that will cause a distortion of the plasma membrane. That will open up the channel, allowing ions to enter the cell, and once the pressure is gone, they will close back. So as I said before, the membrane potential changes when the permeability of the permeability of the plasma membrane changes, and that will allow the ion to move across membrane. So once the ion move across the membrane, the concentration of the ion inside and outside of the cell will also change. And for that, you can get two types of signals. One is called the graded potential, and in the graded potential, when the electrical activity happens, it will be with a short distance. Okay, it will not go to the long distances. Compare that with an action potential that it can travel to long distances, and I will give you more detail about that. And based on this changing in the plasma membrane, the information can be sent or received or processed. So this is all the information, sensory, motor, all the higher brain function is based on the transmission of the electrical activities.